So in this video, hello everyone, this is LATHA AKULA. In this video, we are going to study about structure of DNA and RNA. In the previous videos, we have completed that. What are the different types of components of the nucleic acids? What are nucleotides and nucleosides? What are the purines, pyrimidines, etc. We have studied about introduction in the previous video. Now we'll see about the structure of DNA and how the double helical structure of DNA was exist and what is RNA. And here DNA. What is a DNA? DNA is called as a genetic material. What is genetic material in all the living organisms? Mostly it is a genetic material. It is a molecular basis of hereditary. It is called as hereditary material. It carries information from the one generation to another generation. So in all the organisms. Hence the DNA is the major genetic material found in almost all the living organisms. But in some viruses, instead of DNA, RNA acts as a genetic material, like some viruses like tobacco mosaic virus, HIV virus, polio virus, measles virus. In all these, the RNA will act as a genetic material. And here, the nucleic acids, they exhibit a wide variety of structures. In all those structures, the most familiar structure is Watson and Crick model. That is double helical structure proposed by the Watson and Crick model is the most familiar one, which we are going to study about that in present video. The Watson and Crick model of DNA. According to this, here Watson and Crick are the two scientists. They studied about the double helical structure of DNA. What is this name of the structure? Double helical. Here two helix are present, right? This is called double helical structure of DNA. So according to this, the DNA exists in double helix. That means two strands were present. How many strands are present? This is one strand and this is another strand. So two strands will form the double helical structure of DNA. And each strand is made up of chain of nucleotides. This is one strand and this is another strand. This is the model and this is the elaborated form. We'll see here. Each strand is made up of a chain of nucleotides. Say this. This is a nucleotide. This is nucleotide. This is form a chain of nucleotide, right? And here, this is the nitrogen base. This is the sugar and this is a phosphate group, and this together form the nucleotide. This is the nucleotide. This is the one nucleotide and this is another nucleotide. Here these two nucleotides are connected by means of bases, hydrogen bonds. Here these nucleotides chains will be connected, adjacent nucleotide chains will be connected with one another by means of phosphodiester bond. We have studied about the phosphodiester bond, right? Here this phosphate group that form the ester linkage with the sugar, with the fifth carbon of one sugar and third carbon of another sugar, it forms the ester linkage that is called as phosphodiester linkage, right? Here each strand is made up of chain of nucleotides linked together by means of phosphodiester bonds, right? Here this chain is connected by this phosphodiester bond, and this one and this one was connected by this phosphodiester bond, like this. These phosphodiester bonds, they connect the adjacent nucleotide chains. And here this is one strand and this is another strand, right? These two strands, they run in opposite direction. That is, they were anti-parallel. What is meant by anti-parallel? You see here, one strand runs from the 5' to 3' direction. Another strand runs from the 3' to 5' direction. See here, 3' to 5' and 5' to 3'. That means two strands run in anti-parallel. They run in opposite directions. That means here what is meant by 5', 3', 3' to 5'? Here in 3' direction, the third carbon of the sugar is free. In the 5' position, what will happen? The fifth carbon of the ribose sugar is free here. That is called as 5'. And here the third position of the sugar is free, then it is called as 3' end. Here these two strands, they run in opposite directions. And here this double helical structures, they were coiled around an imaginary axis. And here These two strands, they can be imagined like a spiral staircase. So in the duplex houses, we'll see the spiral staircases, right? Here, this double helical structure also resembles the spiral staircase. Here, the base pairs are present, right? They will be resembles like the steps of the staircase, and here these two strands which will form the backbone. What is the backbone here? Sugar phosphate is the backbone, right? Here this backbone will act as the railing of the staircase. The steps are nothing but the nitrogen bases, and the railing of the staircase is nothing but the backbone, that is sugar phosphate groups. And here the base pairing is specific. This is a nitrogen basis, right? Here this is the purine and this is a pyrimidine. This is a purine and this is an pyrimidine. Adenine pairs with thymine, guanine pairs with cytosine. Adenine always pairs with thymine, or thymine always pairs with adenine only. Like that, guanine always pairs with cytosine and cytosine always pairs with guanine only. Here between these two nitrogen bases, between adenine and guanine, there are two nitrogen bases were present, and between guanine and cytosine, there are three hydrogen bonds are present. So how many hydrogen bonds are present? The number of the hydrogen bonds is very important. How many hydrogen bonds are present between the adenine and thymine? 2. And between guanine and cytosine, 3 hydrogen bonds are present. And here between these two nitrogen bases, pairing is formed. This is called as base pairing, and this base pairing is specific. That means only adenine pairs with thymine only. Guanine pairs with cytosine only. There is no change. Such base pairing is called as complementary base pairing. That is, they were not identical. That kind of base pairing is called as complementary base pairing. Here, if you see the structure of this double helix, this is the diameter between these two. What is the diameter of this strand? 20 angstrom units. And between these two, between the base pairs, what is the length of these two base pairs is, see here, 0.34 nanometers. What is the length? What is the distance between the two base pairs? 0.34 nanometers or 3.4 angstrom units. 0.34 nanometers or 3.4 angstrom units. 3.4 angstrom units. And here say this is called as a minor groove and this is called as major groove. Here in the helix, per 1 turn, for complete 1 turn, that is 360 degrees centigrade is the 1 turn, right? What is 1 turn here? 1 minor groove and 1 major groove, that forms the 1 turn, that is called as pitch of the helix. What is 1 turn? 1 minor groove and 1 major groove together form the 1 turn. Per 1 turn, there are 10 base pairs are present. How many base pairs are present per a turn? 10 base pairs are present from 1 turn. And here, what is the length of the 1 turn here? Here, the length of the 1 turn is 34 angstrom units. What is 1 turn is called? 1 turn is 1 major groove and 1 minor groove is called as 1 turn, right? It takes the length of this 1 turn is 34 angstrom units, and distance between the two base pairs is 0.34 nanometers or 3.4 angstrom units. 3.4 angstrom units. And here this is a right-handed helix or clockwise direction. How it will be running? It was running in clockwise direction or right-handed helix, and this type of DNA is called as bDNA. What is this is called? This is called as bDNA. So what we have studied up to now, so here base pairing is a specific, and the specific base pairing is called as complementary base pairing. Here one end of the strand runs in the 5' end, and that is 5th carbon of the pentose sugar is free, and other end runs in the 3rd' direction, that is 3rd carbon of the pentose sugar is free. That is only we have studied, right? And what is the diameter of the helix? It is nearly 20 angstrom minutes. And what is one turn of the helix is called? That is called as pitch of the helix. And one turn is called as one minor groove and one major groove that together form the one turn. And that length of the one turn is called is 34 angstrom minutes. And distance between the two base pairs is the 3.4 angstrom minutes or 0.34 nanometers. Here the helix is in clockwise direction or right-handed helix, and this type of helix is called as bDNA. And here there are different forms of DNA. They were named according to the English alphabets, depending upon the number of base pairs and what type of the helix that was exhibiting. There are different types of DNA like bDNA, aDNA, cDNA, ddNA, zDNA, like that. bDNA means up to now we are studying about the bDNA only, right? That is which has a right-handed helix and 10 base pairs per turn was present. And aDNA is it has 11 base pairs per turn, that is called as aDNA. And cDNA means it consists of 9 base pairs per turn, and ddNA means it has 8 base pairs per turn. And if the DNA is left-handed coiling, we have studied right-handed, right? If it is a left-handed coiling, that DNA is called as zDNA. We need to remember left-handed or anti-clockwise direction. If it is, then it is called as zDNA, and it has a 12 base pairs per turn. How many base pairs are present? 12 base pairs per turn were present. And next one is the Chargaff rule. In the year 1950, Erwin Chargaff is a person. He proposed Chargaff rule. According to him, the amount of the purines is equal to the amount of the pyrimidines. That is, A plus G is equal to T plus C. So that is the Chargaff rule. What is Chargaff rule? A plus G, that is amount of purines is equal to the amount of pyrimidines. That is called as Chargaff's rule. This is about DNA. Next one is RNA. So next one is RNA. RNA is usually single-stranded. DNA is a double-stranded, right? And whereas RNA is a single-stranded. But sometimes in some viruses like Rio virus and rice dwarf virus, the RNA may exist in the form of a double-stranded. And here RNA does not follow Chargaff rule, that is amount of purines is equal to amount of pyrimidines, right? That is 1:1 ratio is not followed by the followed by the RNA. And hence because there is no double-stranded nature, hence it will not follow any Chargaff rule. And what is the RNA full form? It is ribonucleic acid, and it is single-stranded. And here where it is found mostly in the cytoplasm, that is outside the nucleus. There are 3 types of RNA. There were messenger RNA, ribosomal RNA, transfer RNA. Messenger RNA is called as mRNA. It is produced in the nucleus and carries the information for the synthesis of the proteins. Hence we are calling it as messenger RNA. It carries the information, right? Hence we are calling it as messenger RNA or mRNA. It was first discovered by Jacob and Monod in the year 1961. And second one is ribosomal RNA or RNA. This is the largest part of the RNA is the rRNA, and where it is found, the name itself indicates ribosomal. That means it is found in the ribosomes. So this RNA is found in the ribosomes where the protein synthesis takes place. Nearly 80% of the RNA is the rRNA. Next one is the transfer RNA or tRNA or soluble RNA or adaptive RNA. So many names are there. And here this tRNA or sRNA, it is called the smallest RNA because it constitutes only 10 to 15% of the total RNA. And it is found in the cytoplasm. Where it is found? It is found in the cytoplasm. And what is its function? Its function is to collect the amino acids from the cytoplasm. It will collect the amino acids from the cytoplasm. Further, during protein synthesis, amino acids are required. Those amino acids was collected by this transfer RNA and carry it to the target place. That is why it is calling we are calling it as transfer RNA. So this is about RNA and DNA. We have completed the DNA and RNA structures, and we have completed the nucleic acids.